Good morning and welcome back to our next session. It is my great honor to introduce Professor P.V. Sunil Kumar. He is a physicist, he's a professor at IIT Madras, he is the founding director of IIT Palakkad, and he is a luminary in his field. He's received several awards and accolades to testify to that, some of which is Professor M.B. Piley Award. He's a fellow of Indian Academy of Sciences and is also a fellow of Indian National Science Academy, among other things. So without further ado, I welcome you to start your talk.
Thank you, and nice to be back here in IMSC. So I was thinking about what topic to present here, and I decided on this topic because this is a problem which I started here in IMSC when I was a postdoc here in 95, 97 time. Okay? And of course, I'm not going to tell you the whole history from 97 onwards, but I just tell you something which I'm doing recently in this area. So, so the problem which we were interested in from that time onwards is actually sorry before I go that this work which I'm presenting is done with my students, former students, TV Sachin Krishnan and Ramakrishnan, and my collaborator John Ibsen, and long-term collaborators both John Ibsen and Madan Rao. In fact, this was with Madan with whom I started doing this kind of problems here in math science in 95. So the problem which we were interested is in understanding. So this is a cartoon of a cell here, and as you can see, there is many compartments inside this cell, and this is the outer plasma membrane, and then there is this many compartments inside the cell. We call them organelles, right? And these organelles are also separated by a membrane, okay, a membrane, and so is the cell itself, right? So there are plasma membrane, and then there are these organelle membranes, which is thing. And these organelles, if I take one of them here, the Golgi apparatus, one of the organelles, and you can see that they have very ramified compartments, and they have, but still they have a specific shape. So the shape of these organelles are actually highly conserved across various cell species. And when the cells divide, these organelles disappear, and then they reappear again in the daughter cells. So the question was that like how they maintain their shape, how they first of all form, and how do they maintain, because they're also very dynamic. There are things going into the vesicle, into this compartments, like in the form of these transport vesicles, and there are things going out of it. Okay, so there are a lot of activity happening there. Things come in and things go out. Okay, but still they overall maintain a very they're in shape, right? Okay, so that's a question which we were trying to answer. So what are the physical mechanisms that might be responsible or that can play a role in determining that? Okay, so we know that as I said, these membranes have a very complex composition. Okay, and there are also as I said, steady flux of material in and out of these compartments. Okay. So these membranes do many things. Okay, they host proteins and they compartmentalize the cell. They also host protein machinery for various jobs and the thing. Okay, and then they also do a lot of sorting of proteins and things like that. So that is a very active system. Okay, so now the central issue here is to explain the morphology, and the chemical composition of organelles as a consequence of this network of forces and involved in the transport processes. Okay, that is something which we want to really address. Okay, so now molecular biology, as you know, is a very advanced field. Okay, and so a lot of information on what are the components which are involved, what are the players involved are already known. Okay, that's available, and people can do very, very sophisticated experiments to knock out proteins and add proteins, etc., modify proteins, etc., to know what is the effect of that. Okay, but what we still don't really understand is how do these elements, which are responsible for maintaining certain things, actually do it. Okay, and that is that is something which is we still don't have a good understanding of. Okay, and that is something which we were interested in looking at. Okay, so now the question, as I said, we want to understand how these organelles maintain their shape. So now, what do we know about that? We know that there are certain factors we know on which how the proteins, for example, are involved in these processes. Okay, and so proteins can actually sit on this membrane. And if this protein has a particular shape, okay, or it can sit on this membrane and take a particular shape. Both are possible. Sometimes proteins do have a particular shape, and then they can force the membrane to adapt to it. Okay, or they can bind onto the membrane and undergo a shape change. Okay, and then that can change the shape of the membrane. Okay, or they can tether membranes to the cytoskeleton. Cytoskeleton is another set of protein network. Okay, a filamentous network which is inside the cell, which is like a scaffold of the, if you want to have an analogy. Okay, for a tent, scaffold for a tent kind of thing. So then you can have these organelles being tethered to those poles of the tent, and so that they can maintain a particular shape. But it's hard to imagine how such a thing would work when it has to also have to be dynamic, and it has to undergo all this transformation when when the cell is dividing, etc. Okay. Some proteins can then organelle shape by stabilizing the morphologies through assembly, right? That is, these proteins can, not individual proteins can assemble, okay, and then dock onto a membrane, and then you can do a shape change, okay. Or they can also there can also be regulated fission fusion. That's something which I will talk about in a little detail if I have the full time for it. So regulated fission fusion of membrane can affect the organelle shape, and that is something. Okay. So okay, and now so two questions which we want to actually look at in this talk, if I have full time to do it, is to address these two things. Is that regulated fission fusion of the membranes, how that affect the organelle shape. Okay, and the next is curvature fluctuations due to protein binding and unbinding, how that can affect the shape. These are the two questions which I probably want to look at in this talk. Okay, so what we know is that just having a lipid membrane, okay, as I said, these membranes which are dividing the organelles, okay, or separating the organelles or the plasma membrane itself, it's a fluid membrane, okay, and made up mostly of lipids, and then there are proteins on it, okay. But what we know is that only a lipid membrane alone cannot give you such very high curvature shapes, okay. So that is something which we all, I think, they're kind of recognized today. Okay, so now the question is, how do I going to model, right? So this thing. So I'm going to talk to you about a computer simulation of these things, so with a model, okay, for a membrane, and that's the purpose of this talk. Okay, and that is so for that I want to model the membrane. So membrane, as I said, is a two-dimensional sheet, okay, embedded in 3D, right? So it's a fluid sheet. Okay, that's very important. It's a fluid sheet which is embedded in 3D, and so that is what I'm trying to show here. Okay, that is a lipid membrane, okay, and you can have a protein sitting on it, okay, and this is a fluid, which means there is no shear modulus, okay, things can diffuse freely inside the surface, and it is embedded in 3D. So the classical Hamiltonian which people use for this, the free energy people use, is actually this one is called the Helfrich-Hanham Hamiltonian, okay, and which has a term which is coming with the mean curvature of that surface, okay, being embedded in 3D as a 2D surface. The mean curvature is given by as 1 over R2. Okay, and then there is a Gaussian curvature which is possible, right? When there's Gaussian curvature given by that, and then there is a area change, surface tension, which controls the area change. So parameters here would be actually kappa, sigma, and this Gaussian modulus. Okay, and in the whole talk which I'm going to talk about, I will not use this term because I assume that the topology of the whole the object I'm looking at remains spherical. Okay, and hence there is no change in the. This term does not contribute. That's an assumption, assuming that whatever I'm going to talk about is a uniform. These moduluses are kg and kappa or etc. are uniform across. Okay, so that is something. So I'll be talking about, and then you can take this model and then you can sophisticate it. That can have. Helfrich Hamiltonian can be modified to include inclusions and have a nematic field, etc. on top of it, okay, orientational field, okay, and this orientational field can then give you very different shapes, etc. But I will not talk about that either here. Just to show you that this model can be then further expanded. But these are continuum models. And what I want to talk as the problem with working with these models is that you need to parameterize the surface to do anything, right? Now, parameterization of the surface is difficult when it doesn't have any particular symmetry. Okay, so it can do it for a spherical surface or for any axisymmetric shape. Okay, for a general shape like what I showed you in the organelle stake. Okay, it is very difficult to do it. So we switch to doing some simulations. Okay, I will come to that model. So before I go into the simulation model, let me tell you what I am trying to actually model here. Okay, there are three things which we want to model. Okay, one is that in a vesicle coming and fusing. Okay, this is what we call a vesicle. It's a spherical surface, a spherical object. Okay, and it can come and fuse. Okay, and when it fuses onto one leaf of the membrane, it can actually induce a change in the curvature. Okay, and similarly, this object can actually have a fission of a vesicle going out, and then it can go back to that. You can have a protein, which I will show you a cartoon here. It can bind onto the surface, and it can take a particular shape, and then that can give a curvature here. And when this protein is unbound, it can go back to that. Okay, or it could think about another protein which is embedded into the membrane. Okay, and this protein, when it is, for example, an ion channel, when it is open, it can generate a curvature, and when it is closed, it can go back to that. So these are the fluctuations you want to look at, this up and down going that, and they are all non-equilibrium processes, because these are all involved consumption of energy, a change of chemical energy into the use of chemical energy to actually make these modifications. So these are all non-equilibrium processes. Okay. Okay. So let me first focus only on these two things, okay, where there is no area change. I'm talking about some protein binding onto the surface and inducing a curvature, and similarly a embedded ion channel being active and inducing a curvature, okay, or back and forth. Okay, so there is no change in the area of the system. Okay, so now the question is, now I'm going to model this. Okay, so I'm going to model this is using the triangulated surface, and this is what I was saying about the connection to IMSC. This is where I started this particular triangulation model. I actually did this first when I was postdoc here. Okay, and this is the whole surface, now a 2D surface is now completely triangulated. Okay, now it's a fisherman's net. So first you start with the fisherman's net, and you say that now how do I remove the shear modulus in that, and for that I would need to remove these connections and then reconnect them all the time. Okay, so I have a dynamic triangulation. Okay, so I keep doing that by just taking a bond here. For example, I can take a bond here and then flip it to this direction, and I can keep doing this. Okay, and when I do that, I change the surface, the curvature. Okay, so it has to be accepted with the Boltzmann weight. Okay, so this is a thermal fluctuations. Okay, so a thermal fluctuation induced curvature change will come from this bond flipping and also from vertex move. Okay, both vertex move and bond flipping can give you that. You can also have things sitting on it. On these vertices can be decorated, and here I'm showing you a dark patch and a hashed patch. Okay, and now these things can then diffuse freely on the surface, and that is facilitated through a Kawasaki exchange move. Okay, you can actually exchange this, and again, a curvature change would be there depending upon is there a preferred curvature different from between these points and these points, etc. Okay, so you can assume this dark patches have something where the protein is sitting, and the hash patches have where there is no protein sitting. So the preferred curvature at this point would be different from this curvature. Okay, so then you can have some energy change as you flip these things also. Okay. Okay, and these points can also then interact. So you have a two-component system where you can actually have these points interacting, and then you can incorporate that also into this model. Okay, so now, as I said, this could have a different preferred curvature from this, and that we denote by a term like this. Okay, and this I call it a spontaneous curvature of that particular protein or in the model with the decorated points. Okay, and then this phi can take +1 or -1 value depending upon whether it is dark or hashed. Okay, and then depending upon that, this curvature the preferred curvature could change. Okay, that's how we put it in the model. Okay, so that's what I said. Okay, and then you can have interaction between these points, and that is given by usual Ising-like term. Okay, that is what we have. Okay, so now with including all of that into the Helfrich Hamiltonian, and in a discrete version of that, you can write it like this. Okay, now H is the mean curvature at any vertex. Okay, and so this is the preferred curvature. okay, of the vertex depending upon the value of phi, okay, and this is the interaction between the inclusions, okay, and that is a volume control of the volume, the whole, because it's a closed surface and I control the volume with a delta pressure delta P. Okay, that's what we can have. Okay, so now, so this is the Hamiltonian which we are going to simulate, and remember that I know how to compute the curvature at any point, and I won't go to the details, but I can even compute the curvature tensor at every point given the triangulated surface. Okay, and but here we are using only the mean curvature. But if you want to put in the Gaussian curvature, it's possible to do it. Okay, so now, oh, sorry, there is something got shifted. Okay. Okay, now the DTMC here is dynamic triangulation Monte Carlo. Okay, now we have the, okay, I think this I already explained, so you can do. Okay, now the point is that this phi I said is can actually go from +1 to -1. Okay, and that is like a protein binding or unbinding. Okay, or a embedded protein opening and closing and doing a conformational change. Okay, now that I said is going to be a non-equilibrium process. So it's a rate. Okay, and that rate support as this. So plus going to minus is that rate, and minus going to plus is this rate. Okay, I will not have time to explain all the terms, but you just, for the time being, you just assume that there is a particular rate at which things happen, and this happen in a non-equilibrium way. So it is, and you can show that the whole cycle is, you can go to a steady state and then etc. You can show, but I will not go to the details here. Okay, so now these transitions here actually create and destroy curvature. Okay, and then in a non-equilibrium fashion. All right. Okay, now this was published some time back. Okay. So now let's look at this case. Okay, now I'll just show you one or two movies, okay, of that. Okay, so this is parameters which I'm using here for the transition, etc. Okay, and then you can show that these red points are the points which actually create curvature, and I start with a spherical vesicle, and I have this transition which is happening, which is points come and go. These red points are the curvature inducing points, and then so they fluctuate because they are there and then they go away. They are removed, okay, and then you can see that they take a particular shape which is different from a spherical shape, and that's a steady state shape. And depending upon what is the value of the spontaneous curvature which I am using here, this is 0.7 or 0.9, I can get different shapes. Okay, so you can generate a whole zoo of shapes in this process. Okay. And so that is, and then what you can show also that when these are steady state shapes, and you can show that when the activity is stopped, okay, and then it can go back to a spherical surface, and you can see that this is a rate at which activity, and the activity stops, it can go back to a spherical shape, and it's quicker here. And what is the difference between these two are that here the red points don't interact with each other, here the red points do interact with each other. And when they interact with each other and then attractively, okay, so there should be actually a phase separation, but once you have the activity, that phase separation is suppressed. Okay, and that is another point which I wanted to show. Okay, so you saw that when you actually have the activity, it goes from a spherical shape to a different shape. Okay, and that you can actually kind of quantify by looking at the volume of the steady state object, or reduced volume with respect to the volume of the sphere. Okay. Remember, in this whole process, area is not changing. There is no change in the area is constant. Okay, but the volume do change because the pressure we have put as 0, delta P is 0. So volume, we allow for volume change. And now this is the relative volume of the two, the thing for different rates of activity. Okay, different rates of activity, and this is the equilibrium fluctuation which you see here. Okay, and then you can see that as you have activity, okay, as a function of different values of this spontaneous curvature, the preferred curvature which it likes to have, okay, you have this transition to a different shapes, okay, so from a spherical shape. Now, the interesting point here was that, if you, now, as I said, it goes to a different shape which is different from sphere, okay, of course there is now the volume is lower, okay, but then you can slowly jack up the pressure inside the vesicle, okay, and inflate it back, keeping the activity on. Okay, you keep the activity on and the steady state, and you jack up the pressure inside, okay, and then it can go back to the spherical shape, and you can then measure that. What is the pressure required to keep it back to to take it back to the spherical shape, and that is what actually we're showing here. Okay, you can actually for different activities and different C0 values, this curve of the relative area, the volume, okay, as a function of pressure can actually be collapsed onto one single curve. Okay, you can get a data collapse. Okay, and then this additional pressure which you added, okay, you can actually look at that, you can plot that as a function of these parameters, which is kappa is our bending constant, C0 is the spontaneous curvature, epsilon is the activity, and R is the radius of the vesicle which I started with, and we can see that they actually fall on one linear curve. Okay, so basically saying that there is an effective pressure which is coming from the fact that there is this fluctuation in the curvature which is induced non-equilibrium way. So that is what this non-equilibrium fluctuations are doing, actually inducing a pressure, okay, a negative pressure, and that allowing it to collapse. How much time do I have? Only 3 minutes, okay. Yeah, okay. So maybe, okay, just to do justice, I also wanted to just show you that our model can then be extended to include area fluctuation. And this is the one which we have done recently. So this has been troubling us for a long time. How to this shapes with triangulated surface with constant area was actually relatively easier to do. But we wanted to also have area fluctuation, because when you actually add a vesicle onto the surface, you actually change the area of the thing, or the local density, the projected density actually changes. Okay, and we wanted to introduce that into the model. Okay, and that is again this addition. So that we do now by actually adding a vertex, okay, or removing a vertex. So you can take a polygon like that, and then you can add a vertex, okay, or you can take a polygon like this and then remove a vertex and retriangulate the whole thing. Okay. So now this has to be done, and this triangulation and retriangulation of this thing as a function of as you add and remove vesicles into that. So adding and joining of a fusion of a vesicle or a fission of a vesicle is now modeled as a addition of a vertex or a removal of a vertex from the thing, and that's again done with a non-equilibrium rate. Okay, and at this rate. Okay, so you can do that. And the other part of the Hamiltonian will remain the same, except that there is now an area, and there is a little bit of technical details of how you can do that. I will skip this, okay, in this part. Okay, just to say that now this is our Hamiltonian is now this. Okay, so there is one extra term which comes in from the fact that now there is a density, projected density is now changing. Okay, otherwise the whole thing remains the same. Okay. Okay, so now so going to that, now I just wanted to show you that again, this is some technical detail. I want to show you that what happens if I do this. Okay, so now I could have this preferred curvature to be as a one point which I want to point out is that earlier I showed you that there is an effective pressure which is coming in. Okay, as you add and remove the proteins, and that effective pressure is proportional to this spontaneous curvature square. Okay, so when you actually if the preferred curvature is negative or positive, the preferred mean curvature is negative or positive, it doesn't care. Okay, it's the same pressure which is induced. Okay, and when the moment you have area fluctuation, what you see that it becomes sensitive to the sign of the curvature. Okay, sign of the curvature which it has, and there is a reason for that. Okay, and that is one thing which I wanted to show you here is that when I add here. Okay, when I am adding a vertex, I am adding always from the outside of this vesicle. Okay, and then so what we are adding is always a positive curvature. So if the preferred curvature is also positive, then that vertex that added point will actually, the added object will actually sit there for some time and it diffuses very slowly, and slowly it will relax. So there is a relaxation time for that thing, and so there are many time scales here. One is the relaxation time of that curvature, and another is a relaxation of the diffusion time scale for this vertex to move. Okay, and then there is an activity time scale, at the time scale at which you add and remove, right? Okay, so now if I do a negative, if a preferred curvature of the added point is negative, then you can see that I added it here, okay, with the positive curvature added outside, because I'm adding from outside, so I'm always adding positive curvature, but then this vertex will immediately diffuse out. Okay, so the diffusion is faster, diffusion of the added protein or what object, which is faster if the preferred curvature is negative, and if it is it's slower if it's positive. Okay, so this is so now this makes a big difference, and what we see here is that what is there. So if I look at again the relative volume, okay, reduced volume. Okay, if I look at this, and you can see that this all the blue curves are here for negative curvature, and all the red points here are for the positive curvature, and you can see that they have completely opposite effect. For slow rate of activity, the negative curvature, slow rate, you have a deformed vesicle, okay, and it goes back to a full vesicle, okay, as you increase the activity. but for the positive curvature, it's exactly the opposite. Okay, the positive curvature, it behaves similar to without area fluctuation, but with negative curvature, it moves differently. And the reason being that in one case, because when a high rate of activity, you tend to remove the points which are added, okay, because only the points, the red points which I showed you are the one which is kind of removed, okay. At a high rate of activity, you are doing addition and removal almost at the same points, and so the curvature fluctuation is similar, while for negative curvature, you are actually adding a positive curvature and removing a negative curvature. So that then that goes to two different things. And you can see that the shape changes here. I've shown you in the thing. Okay, so I think that is this is again showing a movie of the thing. This is for the positive curvature. Okay, as you increase the activity, and you can see that for low activity it is a sphere. Okay, as the activity goes up, it goes into a non-spherical shape. And yeah. So okay, so because I don't have much time, so I will probably skip that. You can see that it goes into this, and then similarly for here, it's low activity. It is the spherical, this kind of cylindrical shape for negative curvatures. Okay, and as you increase the activity, it will come back to a spherical shape. Yeah, so I think that is all I wanted to say. Okay, so the main reason for this, which as we understand, is that the addition of material is all from outside. So there is a symmetry breaking there in that sense. Okay, we are always adding a positive curvature, but then added point or added vertex or added protein has two different preferred curvature, positive or negative, and that makes a big difference. Okay, and you can actually understand this using these three different time scales in the process. Okay, so I think with that I will just probably stop it here with my concluding points. I just wanted to say one more thing, is that actually we can also do you can take this spherical shape, okay, and do a linear stability analysis using the continuum model which I talked about earlier, okay, for density fluctuations also, for projected density fluctuation, and show that the spherical surface is actually unstable. That we could do. The difficulty is only that what we do as a continuum model and what we do as a discrete model for simulation, matching the parameters are difficult. Okay, because so that is the thing. So, but in the paper we actually present this density as this linear stability analysis also. So I'll stop it here, and then if there are any questions, I'll take it. Thank you. I hope.
Okay then, thank you so much.
Thank you.
We'd like to give you a small memento.